So today's class is about differential leukocyte count. So what is differential leukocyte count? Previously you have counted total, na? So this is about differential. Means? Okay, so see, in the introduction, what is differential leukocyte count? It is used to determine the relative number of each type of WBCs in the blood. Okay, so each type of WBCs will be counting and it will be expressed in percentage. Okay, so this is the introduction, and there may be certain diseases where you will get increased number of WBCs. Specifically which WBCs, okay, will be knowing in this class how to do the experiment, and in which cases it will be increased, single or double. It may increase, it may decrease also. Okay, so our aim, what is our aim? To determine the distribution or relative percentage of WBCs in a peripheral smear. Okay, so what are the apparatus we'll be needing here? So we need a microscope, 3 to 4 glass slides, then a sterile needle or lancet, a cotton swab, spirit, the Leishman stain. So here we'll be using Leishman stain for staining the differential count. Okay, then distilled water. Okay, then staining rack where you will be keeping it after doing the staining, and then you will be staining. Okay, and then the oil. This is cedar wood oil. We are using here cedar wood oil. Okay. So what is Leishman stain? So you know it from before, right? You have used it or not? Where you have used it? Smearing, do you use? Oh, this is for differential count. Okay, so here we are using this Leishman stain. So this is the simplest and the most precise method for staining blood for diagnostic purposes. So this is a type of Romanowski stain which contains both acidic and basic dye. So which is the acidic dye here? Eosin. So what this acidic dye will be doing is, it will be staining the basic particles, okay, like the granules of the eosinophil, the RBCs, okay. And what is the basic dye here? Methylene, yes. So as this is a basic dye, this will be staining the acidic charged particles or the which are negatively charged, okay. So the granules of present in the cytoplasm, the nuclei of the leukocytes, the granules of the basophils, okay. What will be the color? Yes, blue, white in color. And what will be the color of the acidic dye? Yes. So those particles will be pink in color, okay. And one more. This is acetone-free, water-free, absolute methyl alcohol. So, is there any purpose why we are saying acetone-free, water-free, absolute methyl alcohol? So, for this you have to know what does acetone does to the smear. What does water will does to the smear, okay? Okay, if you use acetone, what will happen? Acetone will cause shrinkage, crenation and lysis, okay, of the cells. And what will water do? If there is presence of water, clumping or rolle formation, okay, this will cause rolle formation. So in the here this methyl alcohol is doing the fixing, okay. So how it is doing the fixing? So when you are taking the blood, there is the freeze fluid, there will be presence of plasma proteins, na? So this will cause precipitation of those proteins, and this will allow it to fix with the slide. Okay. So now you know why we need to have acetone free, water free, absolute methyl alcohol.
So why acetone free? Why acetone?
It causes crenation. It causes shrinkage, crenation, and lysis of the cells. Okay. And also, alcohol preserves the morphology or the chemicals. Okay, chemical status of the cell. Okay. So, what is the principle? A blood film is stained with Leishman stain and is scanned under oil emulsion from one end to the other. As each WBC is encountered, it is identified until 100 leukocytes has been examined. The percentage of distribution of each type of WBC is then calculated. So, when you will be focusing, at least you have to count 100 WBCs, okay, per smear, okay. So, we will be knowing how you will be counting the WBCs, okay. So, knowing the TLC and different count, it is easy to determine the number of each type of cell per cubic mm. Okay, it is always in cubic mm of blood. Okay, so before preparing going for the blood smear, you have to know what are the precautions you will be needing before going to the preparation. Okay, so there are some precautions. So, what are the precautions? See, the slides should be absolute free from dust and grease, because blood will also stick to areas where oil from your fingers have been left. Okay, why it should be free from dust and grease? So this is the reason. Okay, the edge of the spreader. So here you'll be taking at least 4 to 5 slides and one spreader. Okay, the spreader you will be using the width of the spreader, okay, not the length, that portion. Okay, so here there are 4 edges. Okay, so with one spreader you can at least prepare a smear, 4 smears na? So you'll be using that. Okay, so the edge of the spreader should be smooth, not chipped, otherwise the slide would have striations long and across the smear. So this is the reason why we need a smooth and chipped, not chipped. Okay, this will cause striations. Okay, so leukocytes may also be caught in the chipped places and be carried towards the tail. Okay, because this will give you an uneven smear. Okay, the shape of the smear should be a tongue shaped. Okay, smooth and uniform. Okay, when applying the slide to the blood drop from the finger prick, do not touch the skin with the slide. Okay, but not only the periphery of the blood drop. So it has to be. So if this is the slide, at least 1 centimeter, you have to keep a space. Okay, then you have to place the drop. It has to be upwards. Okay, you cannot place it in the middle or in the lower. Okay, so the shape will depend on that. Okay, there is 3 parts, okay, of the peripheral blood smear. The upper part is known as the head, the middle is the body, and the lower part is the tail. Okay, so to avoid taking up epidermal, okay, this is other epidermal squam or sweat. Okay, that is why we don't use touch the finger, only the drop should be touched. Okay, so the other precaution is the blood should be spread immediately after taking on the slide. Any delay will cause clamping of the cells due to partial coagulation. This will give a granular appearance to the blood film, which is visible to the naked eye. So within 30 seconds you need to make the smear. Okay, why you need to make the smear? Okay, so there will be coagulation, na? So if there is coagulation, again the blood will be spread equally or uniformly. So again you will be having errors, and the angle should be 35 to 40, at least 45 degree must be maintained. Okay, the angle of the spreader. So if this is 90 degree, so how will you know which is the 45? At least halfway, na? At least halfway. And which part you will be using? The smooth part. But which part? Yes, the width. Okay, not the length of the slide. Okay, the spreader. So, if the angle is less, what will happen? It will be, see, if the angle is less, it will give a thinner film, and if the angle is more, it will give a thicker film. Okay, so this you have to keep in mind. And the other is the pressure. So you can apply a little bit of pressure. You cannot press it. What will happen if you press it on the striational form? There will be unevenity. Okay, it won't be uniform. So a uniform. So when you will be making the smear, it should be slow and smoothly and uniformly. You cannot rush here. Okay, if you rush here, again the contact of the spreader and the blood, it will be separated and you won't get a proper tongue shaped smear. Okay, the film should be dry after waving it in air immediately after spreading it. So after spreading it, you have to dry it. Okay, so any delay again will cause clumping, also crenation and distortion of the cells. And the other is by turning the spreader over, you can use it to make 4 films. I told na, the spreader, the edge of the spreader, there are 4 edges. The width, if you consider, there are 4 edges. So with this, you can prepare at least 4. So before exam, you make sure there are at least 4 slides. Okay. From why we are taking 4 slides here? So suppose you cannot make a proper smear in a first go. So from the 4 best slide you have to take. Okay, for staining and fixing. Okay, and do not allow the Leishman stain to become thick or dry on the slide, as this is for precipitation of the stain. Suppose you have prepared the slide. So suppose if this is the slide and the smear is, the head is this part and the tail is this part. So you have to give the drops applied in the whole. Okay, from head to tail. Okay, it shouldn't be in a single position. Okay, suppose you are staining only the head, it shouldn't be like that. You have to place it uniformly. Okay, evenly you have to put over the whole smear. Okay, so if you are putting it in a single part, it will cause precipitation. Okay, so you won't be getting. So when counting the cells, use the battlement method. This is to avoid counting a cell twice. So this is a part of the, you have done a zigzag. In case of the WBCs, RBCs, you have followed now that L rule. You have gone from right to left, again from left to right. So this is known as battlement method. Okay. So this method is we are using because of avoiding counting a cell twice. And avoid counting the leukocytes in extreme ends of the head or tail along the edges of blood film. So why we are saying avoid counting the cells in extreme parts of the head, tail or the edges? Why? Why you are saying? So we know the RBCs are plenty in number. So it has a large count. Na? So when you will be staining it, if you are doing the procedure correctly, so the RBCs will be equally distributed through the whole smear. But in case of WBCs, it follows the rule of ununiformity or irregularity. Okay, RBCs follows this rule. You followed now? You are being able to follow me? RBCs are distributed through the whole smear evenly. But in case of WBCs, they are following the rule of irregularity or uncertainty. Okay, so everywhere you will be getting every type of cell. Because suppose you are focusing on the head or on the tail or on the edges, so there are specific areas where you will be getting some certain number of or certain types of cells. Okay. So here we will mainly be focusing on the body, okay, the central part, not on the head, not on the tail, not on the edges, okay. This is other thing. Okay. So we'll be going to the. So now you have known the precautions which you will be taking before starting the experiment. Okay. So what is the procedure for preparing the blood smear? So you have already done this. So you will be knowing it. So first you have to clean the slides. Two to be covered with the blood film and one used as a spreader. So here we will be taking 4 slides, okay, and 1 spreader, okay. Clean the finger with alcohol, allow it to dry, and then prick it with a disposable lancet to obtain a drop of blood. Okay, so you always use a lancet, okay. So why we are saying to use a lancet? Why not a needle? So the lancet is having a certain amount of length or thickness, okay. With the needle you cannot maintain. So the lancet, that is why we are saying. So you remember the previous classes we are saying, what should be the depth of the prick? At least 3 to 4 mm. Why we are saying? It is not possible to maintain the depth of the prick. So if you use this lancet, it will be easy for you to do the prick properly, okay. So that after that only you will be getting a proper drop of blood. So that is the reason. So after that, make a fine touch with one end of the slide with the drop of blood, only a small amount, okay. So here we will be taking a small amount, not a full drop, small amount. So here are 4 slides so at least with 1 drop you should be able to put it in the 4 slides, okay. Where will be putting the blood drop? One side, at least 1 centimeter, okay. So the total length of the slide must be how many? Of the blood smear, what should be the total length of the 4 to 5, okay, 4 to 5 centimeters at least. So then place the edge of the other slide on the surface of the first slide, just in front of the drop at an angle of 45 degree. Draw the spreader back until it comes contact with the blood drop, and push the spreader slowly and smoothly to the other end of the slide. So what I told previously, when suppose this is the slide, and if this is the head of the smear and this is the tail of the smear, so how will you draw it? So suppose you have put the drop here, maintaining the 1 centimeter distance. So you'll be coming from lower portion, okay, from lower portion or the opposite part of the drop towards the drop, okay. Suppose you have maintained the angle and you are going towards the drop, and then you'll be pushing it back, okay. So somebody you might go directly come in contact with the blood and directly you have pushing it. So this is not the way. So you'll be coming from lower portion towards the drop, and then you'll be coming back, okay. Everyone understood this part? You understood? Last bench? You guys? So this is the way how you draw a smear, okay, or prepare a smear. So after that, allow the film to dry at room temperature. That is, the blood smears should be air dry. So you have to air dry it before going for the staining purpose, okay. So this is how, see, at maintaining 1 centimeter at least, we are putting the drop on the near the edge, close to the edge, okay, of the sphere, and then see, this spreader, it is coming from the lower portion, okay. We are going from the lower portion towards the blood drop, okay. After that, upon touching it, you have to wait so that the blood, with the help of capillary action, it spreads, okay. So there must also be some distance at the length, or the horizontal part of the, see this part I am always saying, this part it shouldn't touch totally, okay, it shouldn't go total towards the edge, okay, at least this much distance should be there, okay, on both the sides. You have to wait here, okay, then the blood will be uniformly distributed okay, or equally distributed like this with the help of capillary action, and then you have to draw it back. Okay, from here we are going now. From here we are bringing the spreader here, and then we have to wait here, and then after a few seconds when the blood is spread with the help of capillary action, and then we will be drawing it backwards, okay, or downwards. Okay, see, like this. So is this a proper smear? What is the shape here? Yes, striation. So it has to be a tongue shaped. So for it to be a tongue shaped, there must be head, there must be a body, and there must be a tail. Okay. You have to notice this part. Okay. Many of you or your colleagues are doing mistakes in these steps. Okay. Everyone is following me? Okay. Any question you can ask. See this slide again. This is a proper smear. So there are striations now, and there is unevenity. See, it is not a smooth and continuous distribution. So, so you have to maintain the angle, and from backwards or from the position opposite to the blood drop, you have to come back towards it, and then you have to go backwards. Okay, backwards, not forwards. Okay, this is backwards. So like this you have to go. Okay. So about the staining. Okay. So in staining the blood, put the dry slides on the staining rack. So you have air dry it and put it on the staining racks. Then the blood smear should be stained as soon as possible. Okay, it should be as soon as possible stained. Okay, then carefully drop the Leishman stain onto the blood film, at least 8 to 10 drops until the film is covered. I told na, it should be covered. The smear, wherever it is located, it should be covered. Okay, so 8 to 10 drops you have to give, and then you have to wait for 20 minutes. So during these 20 minutes, the fixation occurs here. Okay, I told na, fixation. What was the fixation? Which dye fixes? Methyl alcohol, and it must be acetone free and water free. So this is why we are not using here. Okay. So now we are not using water here. Okay. After that you will be using. So if you use water, yeah, it won't be fixed. Okay. Or it might the blood cells which we have prepared the smear, it will be washed away. Okay, or it will be rollet formation. Okay, so for here we will be waiting for 2 minutes, and then we have to add double the amount of distilled water. Here we are using distilled water. See, this is distilled water, not a tap water. So in exam you have to use distilled water. If it is not there, you have to ask. Okay, all the precautions you have to see, and you have to be in your table before studying the examination. Okay, if anything is not there, you directly ask, sir, this is not being supplied to us, or it is not available. Okay, make sure everything is present in your table before studying the examination. Okay, so here we can add distilled water. Another thing we can add here is buffered water. Okay, so if you are given buffered water in place of distilled water, it should be in the ratio of 1:1. Okay, not 1:2. So if you are given distilled water, so it will be 1:2 ratio. Okay, so if you are adding the Leishman stain 8 drops, so how many drops you will be adding the distilled water? 8 drops if you are taking. How many drops you will be giving the distilled? Double. Yes. It should be double. Okay. If you are adding 10, then if you are adding 10 drops of Leishman, then so the diluted stain should act for 10 minutes. Okay. So here what happens? Yes. So the first portion why we are keeping, that is the reason for fixing. So this is the fixation time. Okay. The cells gets fixed to the slides, and the next we are using the stain and the distilled water and waiting for 10 minutes. This is the staining time. So here there is a usine and the another one is what? Two stains, na? Yes, methylene blue. So these two are acting now here. Okay. So we are giving water here. So now it will be staining. Okay. So you might see a greenish come here. Okay. After applying the water. Ideal blood stains may look like tongue shape, but what are the characteristics? What are the points? How it should look?
There should not be any striations. When you are pushing, no? If your push is jumpy, then you will find there is thick and thin.
So one, you have to read how a peripheral blood smear looks like. What are the characteristics of a peripheral blood smear? Okay, there are 4 to 5 points. Okay, you read this part and how should a stained blood smear look like? Okay, there what does an ideally it looks like? So it should be at least single layer thick.
Go back. Yeah, these are the points.
So it should be
It should not be very thick or not very thin. It will smear.
so it should be single layer thick. Okay, not a single RBC should be over. Okay, or clumping must be there. Okay, minimum clumping can be, but it should be a single layer thick, and the stain should be not over stained and not under stained.
Single layer thick means if it is thin, 2 or 3 RBCs may be stacked one over another. So it is double layer or triple layer. Single layer means only one cell in that layer, only one cell layer will be there. So that will be thin. So here we will find all the cells are separated from each other.
So why we are saying it should be single layer thick? What is the purpose? No. Why? What is the purpose? If it is single layer or it is clumping, what is the purpose? So because of the distribution is there, it should be uniform distribution, and also to identify. As sir told na, how will you identify with the cells? Mainly you have to compare with the RBCs. So if the RBCs are clumped, if they are overlaying one upon another, then how will you know?
In case of double layer, see, this is the WBC. Another RBC is stacking over the W. Then how will you identify?
So how does an understained smear looks like? How will it look like? The RBCs will be pale in color and the WBCs will be colorless. And what does an overstained smear looks like? The RBCs will be bluish and the WBCs will be deeply purple, okay, or deeply bluish, okay.
When we will be giving you marks, we will consider this one also, whether it is overstained or like this case. So we will distribute this 20 marks in various fractions. So every fraction will be assessed, every part will be assessed.
So you have known the method for counting. Sir has already explained to you. So why we need it? There is a relevance for the differential count. This is to find out whether there is increase or decrease in a particular type of WBCs we see in given kinds of diseases, in inflammatory conditions, in any drug conditions or a drug toxicity. Okay. So you know this why where it is acetone free, it has to be acetone free, where it has to be water free. And what is the fixation time? When it gets fixed, when it gets stained, what is the staining time? Okay.
These are the few things that
you have to know.
Ask you in biopsy.
Okay.
There are a lot of other questions. These are a few samples.
So what is the
so you have to be prepared.
So what is the absolute leukocyte count? What is that? Yes. Total count is also known as absolute leukocyte. And what are the other cells you'll be seeing in the smear? Okay. Other cells? What other cells can you see?
Other cells other than blood cells.
Name any disease condition where we see under a peripheral smear, under a microscope, and what do we see? Any condition?
We told you in Buffy code, other than WBC and platelets, some abnormal cells may remain there. So if we do a smear, so these cells may remain here. These are normal cells. The cells which are normally should have been in the bone marrow, if they may come out under different conditions. They should not come normally, but the abnormal cells are produced there, so they may come out. By seeing their presence, you can identify. You can see, you have seen the RBC shape is changed in some disease. Sickle cell, anyway, so you can see.
Sickles.
Identify them.
Yes.
These are not normal, no?
And in deficiencies also, like in iron deficiencies or vitamin B12 deficiencies, the size, the morphology.
Size of the RBC varies. Macrocyte, microcyte.
Why not tap water? Then the approximate size of the WBCs, you have to compare it with an RBC. That is the reason, okay? It should be distributed equally. The absolute values are more significant than the WBC values. The reason is that the WBC, the DLC may show only a relative increase in a particular type of cell corresponding to the change in the other cell types. For example, a neutrophil count of 85% may suggest neutrophilia, but if the TLC says it is 8000 per mm cube, then the absolute neutrophil count will be this, would be within the normal range. You understand this part? Understand this point? This point? So suppose you are going for DLC and only the neutrophil has been increased here. You have counted with that box, the 10 to 10 box, you have counted and you have found that there is increasing neutrophil count, okay? It came out to be 85%. So what is the normal neutrophil count?
50 to 70%.
Okay. So you know the percentage. So it has increased. Suppose it has increased, but when you will be doing the TLC, okay, it came out to be normal. So it is not suggestive that an undergoing infection or any pathogen has caused, okay? So it can be also normal, okay? So particular type of this, it cannot be a diagnostic. You have to go for TLC, that is total leukocyte count, okay? That has to be increased, okay? Understood now? Okay, so these are the WBCs, 5 types, mainly granulocytes, agranulocytes, you know it, you know this. Then these are the cells. Sir has already described each cell, how it will look, what will be the nucleus, whether it will be centrally, whether it will be lobed or not, and what are the granules, okay? What will be the distribution, okay? So this is the normal neutrophil, what is the size, it is multi-lobed, then it will be light purple, then it has phagocytic action, and the normal range is 50 to 70%. It is elevated in bacterial infection. We will be providing you the slides, okay? So you don't need to write now. Okay, and these are the eosinophils, okay? Sir has already described. 10 to 15 micrometer, coarse red color granules, bilobed nucleus. It helps in defense against the parasitic worms, okay? And mainly elevated in parasitic or allergic reactions, okay? So this is the basophil. The size is 10 to 15 micrometer, bilobed nucleus, deep blue granules, okay? So the granules will be covering the nucleus, so you won't be able to differentiate the borders of the nucleus, okay? So it releases histamine, heparin. It is less than 1%, okay? It is I think 0 to 1%, okay, normally, okay? And this is the largest cell, 12 to 20 micron. So to identify, see, you can identify here now because the RBCs are separated from each other. It is not double or triple layer, it is a single layer thick, okay? and it is separated. So now you can differentiate what is the size of the RBC. Now you can compare, okay? And it is a kidney shaped. See, it looks like the shape of a kidney. It also has a phagocytic bacterial cells and debris and increase in chronic infections like tuberculosis, okay? And the lymphocytes, they are small, large. So this is the size. It may vary. So 10 to 12, 15 micron, slightly larger than the RBCs. The smaller it will be closer, okay? It will be closer to the RBC size, okay? A large spherical nucleus surrounded by thin rim of cytoplasm. There won't be any granules. Normally it's 20 to 40. There are T type and B type. Both helps in immunity. B cells produce antibodies and T cells helps in cell-mediated immunity. Increase in viral conditions and chronic infections, okay? So this picture is available in your book, okay? How does the granules looks like, whether it will be fine or coarse, okay? Where there will be fine granules, in which cell? Yes, and in the other granule side it will be coarse, okay? Then you have to differentiate within the colors also. So suppose here it is brown, reddish brown in neutrophil, and in eosinophil it will be red, okay, brick red, and in case of basophil it will be purple or blue scanty, okay. And this is the granulocytes, okay. So what the other application, or what are the other uses of the blood smear? So we can detect any infection, any inflammation to determine.
We have got listed.
Acha sir.
So use the application of blood smear.
Yes sir. In poisoning, chemicals, drugs, chemotherapy, radiations, okay, and in DLCs also, in leukemia, and allergic and parasitic infections, okay, mainly increase and you can also know gender of the person, okay. There is a percentage and this is the absolute, okay, and these are the conditions.
This is also important.
Where it will be increased and it will be decreased, okay, WBC count. So in case of neutrophil, these are the conditions.
And you will be able to answer in viral disease what are the cells is going to change. Differentiate out of which cells is going to change. In bacterial infection, which one is going to change?
Okay, so in case of neutrophils, these are the condition when it is increased, and these are the condition when it is decreased. In case of eosinophil, allergic reaction, parasitic infection, autoimmune disease, then drug toxicity, stress, okay. For basophils, same allergic, then leukemias, some cancers, then hypothyroidism, and it decreases pregnancies, ovulation, stress, hypothyroidism, okay. In lymphocytes, viral conditions, leukemias, same bacterial infections, and for prolonged illness or AIDS, immunosuppression, treatment with cortisol, steroids, okay. And for monocytes, viral, fungals, these are the conditions, and in bone marrow it decreases, okay. This is the high, okay, and this is the low, okay.
Now what am I doing? I have clean only, just for showing you again. I'm going to name only using a practically there are two wash it with soap water. Okay, but here okay, slice the thing only, it is washed. So I'm going to wipe it. Okay, to look for if there is any presence of pieces. Like from 1 minute you can do this. Okay, suppose I have finished up. And make sure there are no remnants of the paper on the smear surface. Okay. Suppose this is the upper end of my smear in this paper. Okay, these two smears are already clear. Okay. So, which edge was the spreader edge? This one, no? So I don't touch this part. Okay. So this one is the spreader edge. This color se rakha. This one is the spreader. These three slides are there. Now you know how to make a peripheral smear. First of all, you need to give a prick. Who is going to volunteer for the prick? You have that spirit? First, clean your finger. Clean your finger. One view. Clean your finger. What will happen if I make the angle of the spreader more than 45 degree?
Thicker.
At the head end, it will be more thicker. Okay. And what will happen if the angle of the spreader is less than 45 degree? Thinner at the head end, and it will be more at the periphery. The blood cells will be present more at the periphery. That's why you need to make sure the angle is 45 degree. And what if we give a big drop? Are you going to give a very big drop or very small drop? Then what are the changes you can see in making a smear? If you are using a very big drop, what will happen? The smear may not be single cell thickness.
Double layer.
This one. Okay. And also, it will cover the entire slide. You will see. And if it is also very thin, it will cover very small area. Okay. The recommended area covered by a blood smear of the slide is
4 cm.
2/3. 2/3 the length of entire slide. Okay. Now at first we know all the precautions. We already know the precautions. Why will you place that? Why will you give that blood drop? Wherever you are placing that, just like 1 centimeter away from the edges. Are you going to use the first drop?
No, sir.
First drop needs to be wiped out. Then why? Again the same question. There's the bone tissue fluid, the blood cell down there is normal, you can see like more amount of WBCs here. So should not give that. So now, you monitor yourself and you clean your like fingers. Which finger are you going to prick? The non-dominant hand is more preferred. Okay, on the dominant hand. If you are a right-handed person, you will prick on the left hand. Preferably on these 3 fingers. So suppose I am going to prick on his ring finger. Give me your ring finger. Ring finger, okay. Have you cleaned it? You have cleaned this one, okay. Now you are going to prick yourself. Yeah, then prick it.
Manoj, सारे हाथ पे first aid box.
First aid box nice है. Hold कर दो. Now prick it. अरे सुनो ऐसा नहीं. Before pricking, make sure the spirit is dry. His hand was not dry. That's sweat. Why are you sweating so much? You no? 5 minutes time. You have that problem. Only palmar. Ventral. Does it affect your sensation of temperature? Not deep, not that low. That's it. Only air dry. हाथ में कभी फूले जाना क्यों? Your mouth is full of bacteria. यहाँ पे नहीं लेके आना इसको, ठीक है? You may say, okay, my hand is clean, but the air is also full of bacteria. इसको मैंने try किया. Happens like that. थोड़ा bit of this tissue paper. Now? No, not so big. Place it ना. अरे इतना दूर नहीं. 1. Now this one is the smooth. Now you can use it like so. Okay. I am taking. Now you can take like this also. Suppose one is more comfortable like this, so how will we do? You will place it from here, okay, and you will bring it back, and then touch it. Okay, just spread it. Now at an angle of 45 degrees, I am going to do it like this. See? So why are there so many vehicles? Maybe it was not cleaned properly. Okay, this is not perfect one. Suppose I am going to get it here. That's why it asks to clean the slides properly. See, again there are two vectors. I hope so. It will happen here again also. See, no, it is perfectly clean. See the difference? See the difference? Here it is too much blood. Here it is a bit of less blood. So suppose this was perfect blood, but the smear is not perfect here. Why? Bubbles are here. Bubbles are here.
Slide was not
Clean properly. Okay. Maybe there were we were using the tissues now. Maybe the tissue also has some impurities here. Usko lag gaya, and then those bubbles. See here there are no bubbles. It is clean properly. You have cleaned this now. So now what are we going to do? Next step. That right. Okay. Switch follow. That right. कैसे करोगे इसको? Like this. Remember this is the upper part. Okay. I'm going to that right for 2 minutes. Till it becomes dry. So this is the difference.
Good. They have experienced it, no? What happens to why it happened?
There is impurities.
impurities and the side was not clean.
And these are actually not clean properly. They are greasy. The surface is greasy. Here is the same.
Can you do this?
But you see this? You see this?
Blood flow come in, but the slide is perfect.
Yeah, this one is very good one.
Yeah, very good.
Very good one, this one.
You see how
Not too much, not too less, and the slide is clean. That's it. So you will carefully do it. Cleaning is very important. And how much blood, amount of blood is also, how much blood you are taking. This should not be a very big drop.
important is
Okay?
she is very smart.
That's why she is in my class.
अच्छा समझ रहा हूँ इसलिए हाँ and also perhaps was good but he made a stupid behavior on but I forgot हाँ ये किसी का पास था so it is dry now what is the next step? What is the next step? You're going to place it on the rack. Which rack?
Staining red.
Staining rack. Okay. You should know, you should remember this thing that this part was the upper. The smear is here only. The smear isn't here. Okay. Some people do the mistake, they don't know which one is the upper part and the stain here only. And it will not be doing anything. It will not be any useful to you. Okay. So, I'm going to place it like this. Here only. Now what are you going to do? Add what? Leishman stain. Bring me the Leishman stain. It has dried completely now. Are you sure? It has dried now. You should remember that. Okay, make sure it is dry. How it will be dried? By air dry. Suppose I place it here. Now, this is Leishman stain, right? Are you sure? Sir is told. Okay. Suppose I am going to place it over all of the smear area. How many drops?
8 to 10 drops.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10. For how much?
20
I should
minutes.
need to at
2 minutes.
what is this time called?
Fixation time.
Fixation time. This is not staining time. In this 2 minutes, what will happen? The smear will be fixated. Who will help in the fixation?
Methyl.
Huh? Yes, you are right. Acetone free methyl alcohol. What? Acetone free, water free methyl alcohol. Yes. Why? Why it is acetone free? Have you seen the time? Time देखो कोई? It is already 15 seconds past. Okay, count that. Okay. Tell me when it's 2 minutes. In 2 minutes, I am going to say why we use acetone-free methyl alcohol. आज क्लास में पढ़ा है ना सर ने? हाँ तो why it should be acetone-free? Because acetone causes what? Lysis of the cells.
Lysis.
That's why we need acetone-free methyl alcohol. Methyl alcohol causes the fixation of the cell. These 2 minutes are required for fixation. After fixation only, then you are going to stain it. Without fixation, you can't stain it. Okay. Why it should be water-free?
So to avoid roll formation.
And it also delays fixation. To avoid roll formation and to avoid delay in fixation. Okay. Next, we are going to use water. Do you have this water?
Distilled water.
Distilled water है क्या हमारे पास? नहीं है ना? So we are going to use this tap water. कितना टाइम हो देखो? 30 seconds. Why are we using water here? Tell me. हो गया क्या हमारा टाइम? हाँ? 15 seconds. 15 seconds more. It's leaking. Just like the information in your brain. हाँ? जो भी डाल रहा हूँ तब गिर रहा है. Pause. How much? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14 हुआ क्या? 16, 17, 18, 19, 20. चलो. Now what will you do? Wait
10
for it.
minutes.
For how much?
10 minutes.
Timer set करो. Remember to test the timer. Now I am going to do the air blowing. Okay. What am I doing now? Air blowing. So that the water is and the stain is uniformly spread. So this is called air blowing. Okay. मैंने इसको air blow कर रहा हूँ. See? Now this is flowing ना? That's why you need to air blow. नहीं तो क्या हो जाएगा? एक जगह पर stain fix हो जाएगा. Whenever you might have seen in the previous classes also, retail classes, the part of the smear becomes too much blue. It should not happen like that. Okay. For preventing that, we are going to do air blowing for how much? Till 10 minutes. Okay. Okay, you do it now. For air blowing, there shouldn't be anything in this dropper.
So
Remember?
sometimes we use that straw to air bubble.
स्ट्रॉ से क्या करोगे? फूंक दोगे? नहीं, don't use. मैं बोल रहा हूँ मुंह यूज मत करो. Don't use your mouth. Okay, only use your mouth where in बाएगा. हाँ, not here. So, why it is acetone free? You all know. What is the function of methyl alcohol? You all know. Now, Leishman Staining is a group of Romanovsky group of stain. It has two components: acidic component and basic component. What is the basic component here? What is the basic component here? बोलो ना. Basic component कौन सा है? Methylene blue. Acidic component कौन सा है? Eosine. Eosine is going to stain the
Basic component.
yes basic granules, and methylene blue is going to stain the
Acidic
granules. So who is going to stain the nucleus? Methylene blue or eosine? Huh? Why methylene blue? Because it is basic. It is going to stain the acidic part, and nucleus is acidic. Why? Because of presence of nucleic acid. Which nucleic acid? Deoxyribonucleic acid, DNA. Huh? समझ में आ रहा है? The basics. Okay. Now, और क्या करेंगे इसको? पानी का फ्लो. Same line and then ठीक है देख रहा pinkish green is there. It is not bluish in nature. हाँ? It has become a perfect line now. Suppose 2 मिनट तक हम इसको ऐसे करेंगे. 2 मिनट नहीं, 1 minute is enough for that. हम इसको ऐसे clean करेंगे. There is much of this bluish tinge. It needs more time. It doesn't need more time. Okay. It is already pinkish tinge आ गया इसमें. Okay. The flow should not be too much. Now I'm going to place it here. Now again. So क्या करेंगे? Always touch like this. Never touch like this or like this. Touch like this only, and then again that till it becomes complete, right? And then you're going to focus under microscope. First low power, then 40x, then oil immersion. In oil immersion only you're going to find the different types of WBCs. Okay? I will be sitting here and I will be showing you how are the cells visible under microscope, 100x. Okay? But first of all, you need to dry it. Okay? One of you needs to dry it. Okay? Chalo. Place that where are you going to find? This is the head and the tail end. The tail end there is going to be more of WBCs. The head end there will be more of RBCs. Wow. हाथ में डालो बस एक सब कुछ रहना पड़ना then you are going to focus just a bit behind the tail end, not at the head end. Head end you are going to see clumps of this RBC. Okay, there will be clumps of RBC where you are going to find very few WBCs. Okay, it will be difficult to identify also. So what you are going to look at just behind the and tail part of
hand wash.
So perfect, sir.
I use some hand wash and cleaned it.
So actually, our head also has some pieces, so that also contributes.
Winter season no
Yeah, tail end. Okay. And यहाँ पे तुम पहले क्या करोगे? पहले अपना low power focus में you're going to find okay, it's the perfect place where I am going to place my oil immersion. Oil immersion is the 100x one. 100x one में हम कहाँ पे वो oil immersion में देखना है, focus करना है for DLC. In low power only you are going to see for yourself. Okay, this is the best way. I have focus it here now. Okay. Now हम इसको change करेंगे. Let's go. Leave that cedarwood oil here. Now I am going to place it under 100x. Now the exact place only I am going to focus. And now know how to calculate. What is this method called? Battlement method. Okay. Like just like zigzag method only, na?
Yes.
पहले ऐसे how many cells you can count for 100 cells. Suppose in 100 cells you get 10 eosinophil. What is the percentage? 10%.
10%.
Okay. So it's easy now. That's why we calculate 100 cells. For 100 cells you can make a box. Okay. And E. Like for small lymphocyte you can write SL. For large lymphocyte you can write
Sir,
LL.
how can we identify that it's a 100 cell?
100 cell. You're going to change your field ना like this. Change your field ना मिलेगा तुमको. फोकस करो तो मिलेगा. Suppose dry हो गया. Okay. Now. तो टच करो इसको. What is this called? Condenser only. And what is this?
Light.
Iris diaphragm. So which diaphragm मतलब क्या है? पर्दा है. Suppose this is the पर्दा. हाँ. अगर मैं इसको पूरा खोल दूँ तो क्या होगा? More light should enter. That's what is happening here. See, more lights are entering now. मैं इसको जब close करूँ तो less lights are entering here. This is the light source, artificial light source. So for see, suppose this is our oil immersion. Suppose this is our telex, right? This is our telex. Suppose in 10x, what will be the position of your condenser? Lowermost part, middle part, or uppermost part? 10x, man. The condenser should be at the lower part, and it should be only like slightly. The diaphragm should be slightly open. Now I am going to focus it. What is this? What I am doing? What adjustment is this? Course adjustment. See, in 10x I have found that. You can see the RBCs. Suppose I'm going to look at a place now. What is this? This is the slide.
Stages.
Very good. Suppose I have to sit here. Now I am going to
Next.
ठीक है next में देख लिया how can you see 40x same only फिर इसके लिए हम क्या करेंगे कोऑर्डिनेशन को थोड़ा ऊपर लेके आएंगे एंड आइस डायफ्राम को थोड़ा ओपन करेंगे change the length to 40x now again I am going to focus bring me one immersion one same immersion one now I am going to place how much drops of course one or two drops why we use the immersion oil
match the different.
how to focus match the different index okay why so that the light can travel through direct from one source to another okay सीधा हमारे eye पे आए so that the light is going to travel through the light medium में तो क्या होगा light medium is air medium is lighter so it will be dispersed the light will be dispersed going to convert the light and going to focus in the convert the light that's why you use this steroid oil or immersion oil. How can you focus in 100x? First of all, by using coarse adjustment, first of all पूरा इसको open करना है, ठीक है? Light source को increase करना है, ठीक है? Full increase कर दिया हमने, and then by coarse adjustment you will make it touch, ठीक है? ये touch हो गया. Now धीरे से हम इसको
Good. Yeah.
What have I done? First I made the oil touch the lens. धीरे से जब हम इसको coarse adjust करेंगे, whenever I see the field, I am going to the fine adjustment. Now see, you can see the cells, and you should tell me which type of cell it is. Which cell is this? Huh? Which cell is this? Violet what? Eosinophil का न्यूक्लियस किस कलर का होगा? What is the percentage of basement in a cell in a 0 to 1%?